Okay, welcome. We are going to talk about early life and prokaryotes. This is chapter 27. Sorry, a little delay here. All right, so chapter 27. Again, Mr. Anderson has some great videos that I will post for you on our website. So the main points from this chapter are the following: that life arose from non-living matter in the very, very early Earth environment. And all of these things fall within the general laws of the universe. There was no supernatural event that had to happen, right? All life descended from these very early Earth beginnings. There wasn't any abrupt change that went from like non-living to living. Things and these characteristics of life were acquired gradually over time. Evidence suggests this. And living organisms, once they evolved, they impacted the Earth's environment by changing those original conditions. So in the early Earth, so first off, our Earth is very, very, very old. It's estimated to be about 4.6 billion years ago. The very first evidence of life are called stromatolites. And you can see them kind of on this rock surface. They are fossils of very early prokaryotic cells, right? Prokaryotic. And 75% of the Earth's history, throughout these 4.6 billion years or whatever, the 75% of the Earth's life was microscopic or unicellular. So these large organisms that we're seeing on Earth that we think dominate the planet are very short-lived in terms of the evolutionary time scale. Right. And so if you look again at these three domains of life, the LUCA, the last universal common ancestor, was about 2 to 3 billion years ago. And that split out at this speciation event here. And then notice again that archaea and eukarya share more similarities to each other than they do to bacteria, even though bacteria and archaea are prokaryotic. So early Earth's history, just a quick overview. You don't have to know all of this, but I think it's so interesting. So the origin of Earth, very, very long ago. Formation of our solar system about 4.6 billion years ago. The first evidence of life, estimated to be 3.5 billion years ago. Those are those stromatolites. Estimated that at 2.7 billion years ago, we had the first photosynthetic organisms in the form of cyanobacteria that then started producing oxygen for the Earth. So that changed the Earth's environment. Only about 2.2 billion years ago did we start seeing eukaryotic cells. Remember that those came about through the endosymbiont theory, where a prokaryotic cell ended up engulfing another prokaryotic cell, which then eventually led to the evolution of mitochondria and or chloroplasts, right? 1.5 billion years ago, we got multicellular organisms. Colonization onto land only happened about 500 million years ago. And then humans, modern humans, are only about 200,000 years old, which sounds like a lot, but 90% of the Earth's existence was aquatic in the past. Okay, so here is the kind of hypothesis that we're working with. It's called the gradual chemical evolution hypothesis. And it's basically suggesting that these early Earth conditions, which had very low, had low oxygen, lots of lightning, lots of volcanic activity, and a lot of UV solar and radiation, right? So the chemical evolution theory really states that the synthesis and accumulation of amino acids and nucleotides, right? Remember what those are. Amino acids will help to make protein. Nucleic acids or nucleotides will form nucleic acids, RNA and DNA. So the synthesis of and accumulation of amino acids and nucleotides came from inorganic molecules, right? Remember that monomers join together then to make these polymers. And eventually we come to the protocell, which is the first cell, which is really thought to be the beginning of life, right? And remind me, we can watch this very short video in class. So in this gradual chemical evolution theory, basically you had these UV, you had lightning, and with that, you could go, you could basically form some of these very early things spontaneously. So for example, the creation of amino acids from methane, water droplets, dicarbon, you could then get organic molecules in the form of amino acids. And then eventually you get chains of amino acids coming together that then get somehow separated by membranes. And then you get this first cell, which is called the protocell. And this is really called abiogenesis, right? The formation of living things from non-living, right? And some of the main evidence from that is this experiment in 1953 called the Miller and Urey spark discharge experiment, where they basically recreated early Earth conditions in a lab. They then gave a spark discharge, which would mimic lightning, and they could see that they could retrieve amino acids out of that. Right, kind of giving evidence to support this abiogenesis or the chemical evolution theory. All right, so now we're going to kind of take a deep dive into prokaryotes. So remember, both bacteria and archaea, those two domains of life, are both prokaryotic. Remember what all cells have. All cells have a membrane, right? Plasma membrane. All cells have ribosomes. All cells have some sort of nucleic acid, and all cells have a cytoplasm, right? What is prokaryotic DNA look like? Remember, that is a double-stranded circular piece of DNA. Prokaryotic cells were the only life on Earth for approximately 2 billion years. This is a very, very, very long time, a lot longer than humans have been around. They're found almost everywhere on our planet. From the deep, dark caves of the ocean all the way into like our fingernails. They outnumber all eukaryotes combined. And only a small number are pathogenic, meaning they're harmful to humans, even though we think that sometimes bacteria are bad, but only some of them are bad. So prokaryotes are extraordinarily successful, probably the most successful organisms on our planet. They are metabolically very diverse, meaning they have evolved to take both organic or inorganic compounds as their kind of energy source. They have huge reproductive potential through binary fission, right? They can evolve very, very quickly through natural selection, but why is it so fast? Because their reproduction time is very fast. Only 20 minutes for a bacteria cell to divide into two. So quick reproduction, meaning that they can evolve much quicker. And they can survive in a huge number of environments, in our oceans, on our bodies, in thermal vents, etc. So if we look at the structure and function of prokaryotic cells, we know that they're unicellular and very small. We know that already. One thing that you don't know yet is that they exist in 3 different kinds of shapes. The spherical, kind of circular ones, so if I had to draw them out, I'd just draw little circles. Those are called cocci, not cockeye, they're cocci. The rod-shaped ones are bacilli, and then there's some that are spiral shaped, and those are called spirally, right? So those are the 3 shapes that prokaryotic cells can be in. They reproduce by binary fission, so no mitosis. Remember, mitosis is really division of the nucleus, right? And since prokaryotic cells don't have a nucleus, they reproduce through binary fission. Some of them, but not all, can move with a flagella. And the reason I bring up the flagella is because it represents a really nice example of an analogous structure, right? So if we look at, for example, a eukaryotic flagellum, those are microtubule based. They're much bigger and they have a whip-like motion, right? But they are used to move cells about or move things over cells. Prokaryotic flagella are much smaller and they have a rotor-like motion. So they turn in a circle. So even though they have both evolved to help move cells, they have very different structures. So they're analogous, resulting from, remember, convergent evolution, things coming together due to environmental pressures. So very diverse metabolism. We have photoautotrophs, which are like the cyanobacteria. These are photosynthetic. You can be photoautotrophs, meaning you need light and some organic compounds. You can be chemoautotrophs, meaning you need some sort of carbon source or use carbon and inorganic compounds. Often this is sulfur, and this is why, for example, a lot of bacteria can live in those hydrothermal vents like hot springs, which smell like sulfur, kind of rotten egg smell. And then you could be chemoheterotrophs, where you can consume organic compounds. And here's just some examples of these. If you're interested in what they are, I can tell you during class. So here's just some more definitions to give you these different modes of metabolism, right? Photoautotroph, chemoautotroph, and then we have the photoheterotroph and chemoheterotroph. So in general, if you have these heteros, you have more, you're kind of consuming things, whereas autotroph, you're usually using some sort of, let me rephrase that. Let me rephrase. So if you have photo in your name, you are using a light source, right? If you have chemo in your name, you are using some sort of organic or inorganic compound. Okay, so many prokaryotes are symbiotic, and symbiosis means that they are living together. So they're really ecological relationships between organisms of different species, which are in direct contact with one another. A cool example and kind of a gross example of that are the biofilms on our teeth, right? So plaque, which I'm sure you guys have heard about at your dentist, that they try to scrape off with those metal things. Those are biofilms, surface coating colonies of prokaryotic cells, but they're living on our cells along our gum line here. And that is a symbiotic relationship. So symbiosis does not mean always that they're simpatico, meaning that they're both beneficial. So we have other words for that. Mutualism means that they're both mutually beneficial, right? And for an example of that is the bacteria living in a cow's gut that helps digest the cellulose of plants. The second is commensalism, where one party is benefiting, but the other is not. And that's the large majority of the bacteria on our body. They're benefiting from from being honest, but they don't really bother us for the most part. And then the third is parasitism, where one organism benefits and the other is actually harmed, right? So this is an example of the bacteria that is injected when a tick can bite you, and this can cause Lyme disease. If you look at the structure of this prokaryote, what is the structure? That would be their spiruli. Excuse me, just a minute. Let me pause my recording because someone is yelling at me. He is gone for the whole day, all day long.
Where's he?
Oh, did you get your ID tech thing from him?
I told you to.
I know, but I didn't do it. Did you get it?
I don't.
Can you check? Okay, sorry about that. So 3 kinds of living together is symbiosis. You can have mutualism, both beneficial, commensalism, one beneficial, and parasitism, where one is actually being harmed. So remember that as we're talking about prokaryotic cells, we're talking about both bacteria and archaea. And just there's one big difference that we haven't really talked about that thus far. So we've talked about histones, how bacteria have no histones and no introns. Archaea do have histones and they do have introns, making them very kind of have some similarities to the eukaryotes. A couple other main differences. Bacteria really live anywhere, and archaea are often extremophiles, living in very extreme environments, making them hard to study because it's hard to reproduce those environments in a lab. Bacteria have only one RNA polymerase. Archaea have many. But one of the main differences on how they're classified, especially in the lab, is that bacteria have something, a cell wall that's made of peptidoglycans, all right? Which peptid kind of means protein, glycan meaning sugars, so protein, sugar, cell wall. And this is the target of many antibiotics. Archaea do not have peptidoglycan cell walls. So if you look, and you may have heard this before, is that many times if you have, for example, a bacterial infection, they'll do what's called a gram stain to help identify it, right? So the gram stain uses that peptidoglycan cell wall. So again, peptidoglycans are a polymer of sugars and amino acids that form the cell wall of bacteria. This is the target of many antibiotics. So if you have a gram negative stain, then the stain is not able to access this peptidoglycan cell wall because it's kind of hidden beneath this outer membrane. If you have a gram positive stain, then the peptidoglycan is right on the surface, and therefore it would, for example, be easier to treat with an antibiotic, right? So gram positive bacteria have an exposed peptidoglycan cell wall, making them an easier target for antibiotics. All right. And then I have some practice questions at the end, but we will stop there. Bye.